Good evening, everyone. So, this week we'll be continuing a lecture series on basement membrane zone. This will be the third part of the series. We have covered the formation and function of desmosomes in the part 1. In part 2, we discussed lamina lucida and hemidesmosomes. And in this video, we'll be discussing lamina densa and papillary dermis, which is just below lamina lucida. Okay. So previously we had discussed the attachment of two adjacent basal cells. Now these were the desmosomes. In the second part, we discussed the hemidesmosomes, the integrins, the plectins, the integrins, the bullous pemphigoid antigen, and a little bit of laminins. Okay, and we had left it at that because we had started the lamina densa. In this video, we'll continue from laminins and we will attach the laminin to the underlying dermis. And this will complete our organization of basement membrane zone from top to bottom. Now, it is very important. Basement membrane zone in itself is a very important topic for the residents. It's a very important short note. And if you understand the organization of basement membrane zone, and why is it important? Because for me it was very tough during residency to remember what is the organization, what are proteins I see when we go from basal cells down to the dermis. And if we know where the proteins is situated, we can understand what will happen when those proteins are destroyed, either by mutation or by autoantibodies. So if those proteins are destroyed, what will be the level of the split? And what would eventually be the clinical features. So if you know the location of that protein, you will know what will be the clinical features. So that makes it very important for us to understand basement membrane zone and the organization of all the protein molecules from top to bottom. And if you go through these videos, these 3 videos, I hope that it will become quite easier to understand and reciprocate that in the exams, because I've tried to make it a very simple lecture series so that the concept regarding basement membrane zone is crystal clear to everyone. So without much further ado, let's start the part 3 of our series. This week we'll discuss lamina densa and papillary dermis. So let's recapitulate a bit. And the first part was about desmosomes. Desmosomes are intracellular adhesions, that means in between two adjacent cells, in which two cells are stitched via their walls, like this. The second part was about hemidesmosomes, which attaches the base of basal cells to the laminins in the lamina densa. So it starts from here. Let me change the color of the pen. So it starts from here, and then you have the plectins and BPAG, integrins, BPAG2, and laminins. Okay. And in this part of the video, we'll start from lamina densa, which is responsible for dermo-epidermal attachment. The main constituents of lamina densa are the laminins. Okay, let's move forward. So this is an electron micrograph of the basal cell, and here you can see these IF are the intermediate filaments. These are the intermediate filaments present in the cytoplasm of the cell. These black hump-like structures are the hemidesmosomes. The somewhat translucent, somewhat light shade area is the lamina lucida. A somewhat darker shade area, you can see the darker shade area, it's lamina densa. And then you have the, overarching or this arch-like C-shaped fibers, which are the anchoring fibrils. These are the anchoring fibrils. Remember in the last video, second part of the video, I told you about the difference between anchoring filaments and anchoring fibrils. Anchoring filaments are here, which are composed of integrins, bullous pemphigoid antigen 2, and other proteins, while anchoring fibrils are collagen in nature. Okay, specifically collagen 7. Now this collagen, the collagen labeled here, is the dermal collagen, that is type 1 and type 3 collagen, which are the major constituents of the dermis. So from here onwards, you have the dermis, and here is the epidermal basal cell layer. So again, the basement membrane zone is divided into 4 zones. We have the basal keratinocytes, then we have the lamina lucida, then we have the lamina densa, and then we have the anchoring filaments. Anchoring filaments are part of the dermis. Lamina densa and lamina lucida is in between the epidermis and the dermis. Okay. These are the placement of antigens. The keratins 5 and 14 are the major keratins in the basal cell layer. In the anchoring filament group, we have the bullous pemphigoid and antigen 1 in plectins, bullous pemphigoid antigen 2 and integrins, majorly the integrin alpha 6 beta 4, and laminin 332 are present in the lamina densa along with laminin 331, 511, but we'll discuss them in detail. And the anchoring fibrils are made by type 7 collagen. So you can see this arch here. These are the anchoring filaments. While the bigger one are the type 1 and 3 collagen, which are part of the dermis. So all the collagen are part of the dermis. So this diagram we have already discussed in the last two videos, starting from the keratin filaments, the hemidesmosomes, the integrins, and the laminins. These are the laminins. Okay. In this part, the third part of the video, we'll focus more on the lamina densa and the dermis. Okay. Excuse me. So in the second part of the video, we left our lecture at the level of laminins and we'll continue from laminins going towards the anchoring fibrils and then dermal fibrils. So let's start a discussion from laminins, which are the major constituents of lamina densa. Laminins are heterotrimeric glycoproteins. Trimeric means there are three parts or uh, three strands in the laminin structure. Three subunits, alpha, beta, and gamma. So here you can see the long alpha chain, the beta chain, and the gamma chain. Okay. Now, in the older classification, laminins were labeled as the order in the order they were discovered. So this is laminin 5. Okay. But in the recent classification, it is labeled as laminin 332. 3 means alpha 3, beta 3, and gamma 3. So, and these are the genes: laminin alpha 3, laminin beta 3, laminin gamma 2. Okay. Now, laminins are tissue specific expressed. That means in various tissues in the body in which you have the basement membrane, you have different types of laminins. In the epidermis, you have majorly 332, 311, and 511. Okay. Mutations in the laminin will cause junctional labi. Remember, we are at the level of lamina densa. Anything that happens at the level of lamina densa and lamina lucida, any mutation in the proteins constituting these structures, you will have junctional epidermolysis bullosa. If there are autoantibodies, you will have MMP or mucous membrane pemphigoid. Initially, it was labeled as anti-epilegrin pemphigoid. Okay. So in the last video, we learned that about 10 to 20% of the cases can have autoantibodies against laminins, while the rest will have antibodies against bullous pemphigoid antigen. Okay. Now remember, uh, all the acquired blistering disorders that happens at the level of epidermis are known as pemphigus, pemphigus foliaceus, pemphigus vulgaris. Okay. If it happens below that, at the level of lamina lucida, lamina densa, and dermis, you have pemphigoid. The disorders that look like pemphigus but are not pemphigus. So pemphigoid, bullous pemphigoid, mucous membranous pemphigoid, cicatricial pemphigoid. Okay. So let's move forward and we'll discuss laminins in detail. Now, as I've said that different structures are there. Laminin 111 means alpha 1, beta 1, gamma 1. 332, 311, and 511 are the major laminins in skin. These are the older designation. These are not asked as of now. And you can see that they are the major constituents of lamina lucida and lamina densa. Now, each laminin has three strands: gamma, alpha, and beta, with the alpha one being the longer one. And then you have the globular part, which attaches to integrins. So, laminins as a protein are the major protein that attaches the epidermal basal cell to the dermis. Okay, it acts like a glue to attach integrins to the underlying dermis. The functions of laminins is to maintain structural integrity of dermo-epidermal junction. Now, what exactly happens is these are the basal cell, these are the hemidesmosomal plaques, these are the integrins, and you have the bullous pemphigoid antigen. Integrins and antigen are attached to laminins. Okay, so this is laminin, a tripod structure. Let me change the color. So these are laminins. Now what actually happens is, this laminin is responsible for structural integrity. That means the solidification of basement membrane zone. It is also responsible for passage of signals. So remember all the signals in the form of cytokines, in the form of growth factors. Okay, how does the skin knows that it has to grow? All the factors, all the stimulations are coming from dermis, from the body. So the signals are going to traverse or to cross this barrier, and laminin being the most common or the most abundant protein in the lamina densa, laminins regulate this passage of factors. Healing because all the factors which will control the healing will go through lamina densa. So first important point regarding laminins is that they control the structural integrity of dermo-epidermal junction. They're also responsible for passage of signals from the dermis to the epidermis. These signals regulate cell division, migration of cells, because remember all the inflammatory cells, let's say you have a lymphocyte here, or let's say you have dendritic cells. Okay, so only these two types of cells are capable of traversing the basement membrane zone in the cases of pathology. If there's a malignancy, then the malignant cells will either destroy the laminins or will use this pathway to go inside the epidermis, which is known as the epidermal transmigration of cells. Okay, or as we say as epidermotropism. So in order for a cell to undergo epidermotropism, like we see in cases of mycosis fungoides, they have to traverse this barrier. So they have to either destroy or use the laminin pathway to go across and reach the epidermis. Okay. Now this is known as outside in signaling because the signals are traversing from outside the epidermis inside the epidermis. They go from outside the epidermis towards inside the epidermis, and this is known as outside in signaling. Remember that inside out signaling is done by majorly integrins. Inside out means the epidermis is sending signals to the dermis, and that is majorly handled by integrins. Let's move forward. The second protein that we'll discuss now is Nidogen. Nidogen is a dumbbell-shaped protein. You can see that this looks like a dumbbell, the one everybody is so fond of using it in the gym. I think I should make a good dumbbell. So the structure is like this. One part is heavier, one is lighter because it shows two globular chains. The old name for Nidogen was entactin. This could be a good quiz question if anybody wants to put. These are a group of proteins. They have high affinity for laminins. So what actually happens is at the third globular area, this acts as a attachment zone for laminins. Now this is not to scale. Nidogen is a very small protein. Laminin is a very large protein relatively. Okay. Now the third part has high affinity for laminins. It also attaches to type 4 collagen. So here you can see this is a type 4 collagen binding site. Either it can bind to type 4 collagen alone or it can bind laminins and type 4 collagen. Now remember, Nidogen is like a connector molecule. Either it will connect the laminins to type 4 collagen. And type 4 collagen is a very important collagenous molecule in the lamina densa. Okay. Now, Nidogen is important for rapidly growing or healing epidermis. The second function is stabilization of dermo-epidermal junction. So what actually happens is Nidogen kind of cements the attachment of laminins to the collagen in the lamina densa and the collagen in the dermis. Okay, so it kind of strengthens the dermo-epidermal junction, and it is also responsible for rapidly growing and healing epidermis because at that time you require good functional basement membrane zone. It can also bind to heparan sulfate proteoglycans. We'll discuss proteoglycans in the next slide, what actually they are. Fibrillin 1 are also additional molecules which are present in the lamina densa just to strengthen the lamina densa molecule. Now, if you remember, something will creep into your mind that lamina densa has so many proteins inside. It has laminins, Nidogen, perlecan, heparan sulfate proteoglycans, lattice of collagen 4, fibrillin, and because so many proteins are present at that level, it makes the layer darker, and that is why it was labeled as lamina densa. While in lamina lucida, all you have are integrins, a part of laminin, and a part of bullous pemphigoid adhesion 2. And because of that, that layer appears lighter, labeled as lamina labeled as lamina lucida. Lucida means lucidum, that means bright, okay, or translucent, transparent, okay, because light used to shine through it, so it was light. The third protein molecules are known as heparin sulfate proteoglycans. They are central core protein with laterally emanating glycosaminoglycans in a bottle brush configuration. Now, what is bottle brush? Let me draw a bit of a bottle brush. So here are the other spikes. The brush that normally people use to clean their bottles from inside. This is a bottle brush. So you have a central core, and this central core is a protein molecule, while these spikes are glycosaminoglycans, like a bottle brush configuration. Okay. Now, heparin sulfate proteoglycans can either self-polymerize, that means they can attach, uh, a lot of bottle brush can attach to themselves, okay, or they can attach to multiple lamina densa components. All the proteins which are present in lamina densa, they can also attach to those proteins. Now, this is a unique feature of heparin sulfate proteoglycans. Perlecan is a type of heparin sulfate proteoglycans, and it is responsible to spot weld laminin and collagen for containing networks. So initially, in the last slide, we heard that Nidogen is the major molecule that attaches. Perlecan is also a molecule that attaches collagen 4 to laminins, and it's a stronger bond. That's why the word spot weld has been used in Bologna and Roots. Heparin sulfate has sulfate moieties in between, okay? Now these sulfates, because it is rich in sulfates, heparin sulfate has an addition, a total of negative charge. And because of this negative charge, it repels other negative charge molecules and helps to restrict the permeability. So cells and other materials cannot easily cross the basement membrane zone. So that is why the permeability is reduced, it is restricted. And there is not much passage of material across lamina densa. Okay, let's move forward. Now we come to the collagen molecule in lamina densa. This is, if I remember correctly, the fourth molecule, the fourth protein molecule. The structure of type IV collagen resembles procollagen, in which we mean to say that it has a lot of amorphous substance, it has a lot of globular elements, it is not very truncated. Procollagen, when it goes towards modification, it forms the collagen, and all the excess material which is not required are removed from the procollagen molecule. But type IV collagen has some kind of this excess molecule still attached. That's why the structure resembles, it is closer to procollagen rather than a formed collagen. Like all collagen molecules, type IV collagen has triple helical structure. That means you have three strands in a helix-like configuration. You cannot see. So you have three strands in a helical configuration. Okay, so that is the basic structure. We all remember that collagen has a triple helical structure. Okay. Now this collagen will have, like every collagen, they have repeating glycine XY amino acids. What are XY? XY are the placeholders in which other amino acids can attach, mostly proline, hydroxyproline. More the proline or hydroxyproline, you have the intervening hydrogen bonds between collagen strands to provide more structural stability. Okay. So what happens is that you have different kind of, it's a triple helical structure. So you have alpha, beta, and gamma chains in this also. The alpha 1, alpha 2 subunit of chains are found majorly in skin. The alpha 5 and alpha 6 are also found in skin. 3, 4, 5 are present in glomerular basement membrane in the kidneys. So we'll restrict our discussion to alpha 1 and alpha 2, 5 and 6. Okay. They have a hockey stick configuration. So if you look at this, it resembles a hockey stick. Okay. I'll have to draw it again because the hockey that they mean here, I think it's more like an ice hockey. Okay. So it's a hockey stick configuration. Can you see this hockey stick here? Yeah. So it's a hockey stick configuration. And four hockey stick configuration type IV collagen. Each of these purple cylinder is type IV collagen. Okay. It's a type IV collagen. And four type IV collagen come together to form this kind of a structure. And this is known as a type IV collagen spider. It looks like a spider. Okay. And this spider configuration is what we usually find in lamina densa. If there's any mutation in the type of collagen, you can have Alport syndrome. Now, Alport syndrome is not necessarily restricted to us dermatologists. Okay, Alport syndrome has hematuria. It's majorly because in the kidneys. So it has hematuria. It has sensory neural hearing loss and occasional eye issues, eye abnormalities. Okay, if there are autoantibodies, then the disease causes Goodpasture disease. Okay, and here the collagen 3 is implicated. So mostly it's an issue of glomerular basement membrane. Okay, so mutation in type of collagen, Alport syndrome, any antibodies against type of collagen, Goodpasture disease. Okay, let's move forward. So we have discussed a bit about, not a bit, quite in detail about the proteins in lamina densa. Okay, so here is the laminin 332. This is laminin 332, and at the top of it, it is attached to integrins and bullous pemphigoid antigen 2. If you want further clarification, see the part 2 of this series, part 2 basement membrane, where we discuss lamina lucida and hemidesmosomes, and you will understand what is the configuration. Okay. It is further stabilized by attachment to laminins 311 and 511. Okay. Perlecan, here is the perlecan, which is a type of heparin sulfate proteoglycan. It spot welds. That means it creates a good bond, good attachment between type 4 collagen and the laminins. Okay. Now, for perlecan to attach, you need to have a gamma 1 chain in laminins. That is why it can attach strongly to only 311 and 511 because the last digit is 1, that means the gamma chain is 1. Okay. Now, this is the type 4 collagen spider. Type 4 collagen spider. The Nidogen molecule further stabilizes this bond. As we have already told you that there are three parts. So the third globular protein will attach to the laminins, while the second one will attach to the collagen. We have discussed that in the Nidogen slide, isn't it? Okay. And this arrangement of molecules like laminin 322, other laminins, perlecan, Nidogen, fibulin, collagen, this is present throughout the lamina densa. Okay, all over the lamina densa in a 3D format. Now, the laminins provide further attachment to type 7 collagen, which forms anchoring fibrils. These are the major molecules forming anchoring fibrils. There are other proteins, but this is a major molecule which kind of loops from lamina densa and then goes again inside lamina densa. Okay, and in between that, you have the types 1 and 3 collagen, which are part of the dermis. And that is how lamina densa is attached to the dermis. And since lamina densa has laminins, which is attached to integrins and bullous pemphigoid antigen 2, this is how the basal cell gets attached to the dermis. Let's move forward. So now we have entered the territory of anchoring fibrils in the form of type 7 collagen. Type 7 collagen are the major component of anchoring fibrils. So what happens is you have let's denote it like a line, okay? And here you have the globular head of the collagen and this you have the fibrillar head of the collagen. This is a monomer. Two monomers will form an anti-parallel dimer. That means the globular head will be at the opposite end. And two dimers, when they form this kind of an anti-parallel dimer, the globular ends will be removed. So let me show you two again. Let's see, you have one collagen, then you have another collagen, then you have bonds in between, okay, and then the globular heads are removed. When the globular heads are removed, it becomes an anti-parallel dimer. Two of those dimers join together, then you have three dimers, many more dimers, and this whole will become a tube. Okay, and this tube is the anchoring fibrils. Okay, so in this electron micrograph, you can see this tube going. You can see this tube coming from lamina densa, going again inside lamina densa. Okay, so these kind of C-shaped or arc structures are found, which are known as anchoring fibrils. These fibrils either loop back into the lamina densa or connect to electron dense elements in the sublamina densa region, which are known as anchoring plaques. Now, it has been said that anchoring plaques are nothing but parts of lamina densa which have dropped off. So, what actually happens is, let's say this is the lamina densa. There's this certain parts which can be either here or here, and anchoring fibrils can also attach to this part. Okay, so it has been postulated that anchoring plaques are nothing but parts of lamina densa which have fallen off from the lamina densa during the remodeling phase of basement membrane. Sorry, let's move forward. Now, type 7 collagen has an affinity for type 1 fibrillar collagen in the dermis. Now, remember, this is this section of a skin. Type 1 is more in the top of the dermis. Type 3 is more at the bottom. So, this is dermis, this is epidermis. So if type 7 collagen has affinity for type 1 collagen will be found more in the papillary region of the dermis, and that is how the anchoring these are the anchoring fibrils, okay, the looping anchoring fibrils, and then these are the type 1 collagen. So this is type 1 collagen. And this is how the basal cell is attached to the dermis. If there's any mutation in the type 7 collagen, you have dominant and recessive forms of dystrophic EB. Now we have entered dermis. So D for dermis, D for dystrophic EB, dystrophic epidermolysis bullosa. Okay. So now we have we have gone out of the junction. So no junctional EB now. It will be more of a dystrophic EB. If there are autoantibodies against type 7 collagen, we have a disease known as EB acquisita, epidermolysis bullosa acquisita. Now remember, a lot of disorders share antibodies. That means there's no particular antibodies. For example, in pemphigus, you have desmoglein 3, 1. Okay, so no fixed antibodies are there. A disorder can have multiple antibodies in the sera. You just have to remember the location of those proteins so that you can find out what are the clinical features of that disorder. When we will learn or when we will discuss the acquired or genetic immunobullous disorders, it will become much more clearer why the specific placement of these proteins are leading to the specific clinical features of those diseases. It will be easier to remember. Okay, let's move forward. Now coming to microfibrils, we have reached the type 4 collagen in the lamina densa. We have crossed that. We have reached the type 7 collagen in the anchoring fibrils. We have reached type 1 and 3 collagen in the dermis. Microfibrils are other protein molecules in the dermis which attaches to anchoring filaments. Oh, sorry, anchoring fibrils. Okay. So these microfibrils are present in the papillary dermis and they insert vertically into the lamina densa. So you have the basal cell, the lamina lucida, the lamina densa, the anchoring filaments. Sorry, again, I'm so sorry, anchoring fibrils. And then you have vertically entering microfibril part, and these vertically entering microfibril part are known as oxytocin fibers. Okay. Inferiorly, that means at the lower end, these oxytocin fibers merge with microfibril elements oriented parallel to the skin surface. So inferiorly, below that, let me just erase everything and draw a bigger diagram for you to understand. So you have the basal layer, okay? You have the lamina densa layer. This is the lamina densa, okay? You have the anchoring fibrils and you have the collagen, okay? The oxytocin fibers enter vertically into the oxytocin fibers. Oxytocin fibers enter vertically into the lamina densa, perpendicular, okay? And it is attached inferiorly to parallelly placed microfibrillar elements, providing further attachment of lamina densa to the dermis. Okay, you got the concept? Now, oxygen fibers have a lot of amorphous component. Amorphous means those components which don't have a form. Okay, and these are labeled as alanine fibers. Oxytocin and alanine fibers are contiguous or in continuation with the elastic fibers within the reticular dermis. So, the most abundant protein in dermis is collagen, followed by elastin. So now we have attachment to collagen via anchoring fibrils, collagen. We have attachment to elastin via oxytocin and alanine fibers. So that is how the bond is strengthened between epidermis and dermis. Lincoln is just one molecule. It is a major component of microthread-like filamentous network within sublamina densa. It is responsible for interconnecting microfibrillar bundles, anchoring fibrils, and interstitial collagen fibers. Just remember that lincoln is just one of the molecules to increase the structural integrity of dermo-epidermal junction to provide more support. Just remember that. Okay, let's move forward. So just to revise, these are just the antigens in sub-epidermal immunobullous diseases. So in bullous pemphigoid, you have antigens 1 and 2. Pemphigoid gestationis, you have against 2. Linear IgA disease, 2. MMP, 2. Majorly seen in about 80% of cases. Sorry. Anti-epigram mucous membrane pemphigoid, roughly about 20% of cases, in which the antibodies are against laminin C32. AB acquisita against type 7 collagen, and bullous eruption seen in SLE against type 7 collagen. Okay, these are the level of hemidesmosomes and lamina lucida. Then you have the lamina lucida lamina densa interface, and then you have the papillary dermal area. Okay, now remember, more superficial the split, the more flaccid will be the roof of the bulla. Okay, so that if the split is occurring at the level of epidermis or at the level of the basal layer of the epidermis, the roof of the bulla or the blister will be flaccid and it will break off easily, and the disorder will present more with erosions, not frank bulla. Frank bulla can happen, okay, but majorly when the patient comes to us, the major lesions are the erosions that you see in pemphigus vulgaris. That's why it's an erosive disease, okay. In pemphigus foliaceus, the split is very superficial. So you see more like crusted scaly plaques because a good amount of epidermis is still left. In bullous pemphigoid, the split is at the level of hemidesmosomes. So in hemidesmosomes, the upper layer is the entire epidermis, the floor is the entire dermis, and since the entire epidermis is present, the roof of the bulla is tense because it has a lot of structure present. And that's why in bullous pemphigoid, the presenting feature are bulla, frank bulla, which have not burst. They are tense bulla. Okay. As we go deeper, as we cross the lamina densa level and enter dermis, any bullous eruption happening at that level will cause scarring because now the dermis is getting damaged. And what is there in the dermis? Collagen. Whenever collagen gets damaged, elastin gets damaged, it is replaced by scar collagen, and scar collagen leads to scarring. That's why bullous pemphigoid patients heal with scarring. Pemphigus patients heal with pigmentary disturbances or hyperpigmentation or any kind of pigmentary alterations. Okay. Remember that sweat gland, the ducts of sweat glands or the acrosyringium crosses lamina densa and goes inside the epidermis. When the damage is severe and at a lower level, at the level of lamina densa and the dermis, the damage to the acrosyringium will block the acrosyringium. Okay, so let me just explain it to you very quickly. You have the basal cell layer, you have the lamina densa, okay, and you have the acrosyringium going inside, not to scale, of course. If the damage occurs at a level of lamina densa and the anchoring fibrils, the acrosyringium will get blocked. When it gets blocked, it will get filled eventually with all these sweat products and substances, and you will have milia, clinically milia. So if the damage is below lower level, lower layers, you will have milia, you will have scarring, you will have tense bulla. Okay, so now you remember, now you get some understanding why different clinical features are present in different diseases. Okay, and if you remember that in bullous pemphigoid you have tense bulla, you will automatically remember that the problem lies at the level of hemidesmosomes. When you will remember that the cicatricial pemphigoid heals with scarring, that means you know that it is at the level of lamina lucida and lamina densa. Okay. Anti-pellegrin, EB equisita. If you've seen one patient EB equisita, you will remember. Okay, and that has significant scarring. Type 4 collagen. Sorry, type 7 collagen. Okay, so now you understand. The deeper we go, more milia, more scarring, more dense bulla. The superficial we go, flaccid bulla, less scarring, more pigmented alterations. No milia, nearly no milia. Okay, and that is how you remember to correlate the organization of basement membrane zone to the clinical features of the disease. I hope I made myself clear. This is a very important point to understand and grasp at the level of residency so that you are able to better explain the pathomechanisms while studying immunobullous disorders. Okay. With that, we have finished the third part of the series. It was a short lecture. I've concluded the entire organization of basement membrane zone in roughly about 1.5 hours. If you go through these three videos sequentially, you will easily understand the organization of basement membrane zone, starting from desmosomes and ending to type 1 and 3 collagen fibers in the dermis. If you go slowly and steadily and make notes, you can at least see. When I was preparing these lectures, the concept of basement membrane zone was now much more clearer to me. Okay, so if at any given point of time you will ask me how is the basement membrane zone organized, I can now easily mention all the proteins in order from top to bottom and maybe functions about them whenever you're going to ask me, isn't it? So if you go through important concepts like basement membrane zone, uh, the keratinization process, if you know what is the normal process, you can easily understand the pathology. Okay, so that makes BMZ very important to understand. So if you know melanogenesis, you will understand vitiligo better. If you know keratinization, you will understand ichthyosis better. If you know basement membrane zone, you will understand pemphigus, pemphigoid, and epidermolysis bullosa better. Okay, so I hope this was useful. Go through these three videos sequentially, and I have a very important announcement to make. I will not be making videos for the next 2 weeks. I have a very important personal commitment next week, and a week after that, I would be traveling. So I don't have time to make videos. There will be a gap of 2 videos, 2 weeks. And after I return back from my travels, I'll start a discussion on uh, autoimmune immunobullous disorders. Okay, so we'll start with pemphigus foliaceus, pemphigus vulgaris, bullous pemphigoid, mucous membrane pemphigoid or cicatricial pemphigoid, pemphigoid gestationalis, paraneoplastic pemphigus. Each of this will have separate lectures and in which we will discuss clinical features and pathology and just a short discussion on the treatment because treatment which should be discussed. I've already discussed treatment quite in detail, so we'll go more in pathology and we'll understand the concepts behind these diseases. If you know the concepts, you will never forget this disease. Okay, and it will be easier for you to explain it to your juniors. Till then, keep watching. Send me your comments, your doubts, any suggestions, any queries in the comment section. It's easier for me to reply if it's in the comment section. Emails tend to get lost. And keep watching videos. Of course, we'll be adding a lot more videos with time. I apologize in advance for the next 2 weeks. I will not be able to prepare since I will be very busy. But after 2 weeks, we'll start with the clinical features of autoimmune bullous disorders. Till then, adios and bye bye and enjoy your weekend. Bye.